An interesting thing about antibodies, when they're at a high enough concentration of an antibody specific for what we call an antigen. An antigen is anything that elicits an immune response, elicits a B cell response to produce antibodies towards it. The antigens are going to be that specific amino acid sequence that's part of a protein that's on the surface of blank. Well, anyways, if you get a high enough concentration of antibodies and you get that concentration right compared to what your antigens are, instead of the antibodies just coating the surface of the bacteria, coating the surface, as we see here, red blood cells, what will happen is the antibodies will bind to their antigen, which is on the surface of whatever, but they start to bind multiple because the antibodies are, as you see in the figure, Y-shaped. And they just start to form bigger and bigger structures as they start to bind and they start to cross-link different bacterial cells or start to link together different red blood cells. What they call this is agglutination. They start to bind, so instead of individual red blood cells free-floating, individual E. coli floating around, the antibodies start to link them together and they start to form bigger and bigger clumps, and the bigger and bigger clumps start to settle out. Precipitation. Blood typing test in the hospitals is based on this. You take different serums. Serum 1 is going to be for somebody who made antibodies to the A protein on red blood cells. Serum 2, somebody who made antibodies to serum proteins on the surface of B, and so on. Well, then you take the patient's red blood cells and you mix it with serum 1, you mix it with serum 2, and you see if there's some sort of a reaction. If there's no reaction in the well that has patient's red blood cells and serum 1, because remember serum was towards A, well, that means there's nothing for the antibodies to bind to, which means on the first surface of the red blood cells, there's no A protein. Same with B. If the patient's blood doesn't show any type of reaction, remember serum 2 had antibodies towards B, well, that means there's no B proteins, or sorry, no B sugars on the surface of the red blood cells. So it's indicative that that patient is O. If patient 2 or B's, another patient's blood reacted to serum 1, how would you know? Well, instead of just a nice, evenly red colored blood drop that's in the well, you'll see this looks like sand at the bottom, where all the red blood cells got bound up by the antibodies and they just started to form bigger and bigger clumps that settled out to the bottom. Agglutination. Well, if they're agglutinizing, then that means the antibodies to glycoprotein A has to have something to bind to there. So that means, okay, we know at least the second patient has A on their surface. Nothing happens in the B well, and that means the patient has type A blood. And you just keep going on and on like that. And that's how you tell blood type. Wheel Felix, which is for rickettsia, I believe, or yeah, can't remember. Oh. Same thing. You go through and you do the different tests. See, serum taken from a patient or an animal that had the infection. So we know there's specific antibodies to our infectious agent. Add in blood. See if there's any type of reaction. If there's a reaction, that means then the antibodies had to have had something to bind to. Agglutination tests. Gotta love them.